Hey there, Nishi.
Hi,
How you doing?
I'm doing good. How are you?
Good.
Morning.
So I'm sorry I didn't get that. You know what happened? With the last recording, I um uh, I paused it during the quiz, and I forgot to unpause it after the quiz, so there was no recording. Here I am. I've been doing Zoom for a full year, and I'm still making mistakes. Oh well.
That's okay. Yeah, I panicked a little because I was seeing that recording, and so I'm an audio learner, so I go back. So I like for some classes it doesn't work, but then sometimes Zoom recording really helps me because I can go over and listen to it again and again. But I
Yeah.
went to that recording, and for 15 minutes I was like, I escaped, the quiz part, and I was seeing like the answers, and I was like, that's it. Like, where's the end of the recording? And I was like, oh shit, it's not there. I was trying to figure it out before messaging. And then I asked one of my friends, like my lab partner, like, she's in the same class. So,
Uh huh.
if she's like, no, I don't know. I think he must have forgotten or something like that.
Yeah. Yeah. That's what happened. I screwed up. So anyway, do you remember where it starts? Does it start at the JAK-STAT pathway?
No, it is, I think it's the nitric oxide one. Yeah,
Okay. Okay, we
yeah,
can do that.
that lecture. Yeah.
All right, let me just pull that up.
Great, you're recording this. I want to have this recording.
Yes. I'm going to post it so everybody can see it too.
Okay, perfect.
Okay, nitric oxide, one of my favorite molecules. So let me stop my music here. Listening to a little light Chopin this morning. Turn that off. Okay. So yeah, I really like this because I really like nitric oxide because when I was in grad school, I worked with a guy whose advisor worked this whole system out. So I like my intellectual grandfather was the person who did this. So anyway, I start out talking about my background's cardiovascular pharmacology. So the reason why nitric oxide is important to me is because it's used in nitroglycerin, which is a drug that's used to relax the veins going to your heart, these big veins. And it helps to relieve angina. So angina happens when you've got a narrowing of the arteries that lead to the heart muscle. And so when you exercise a little bit, you don't have enough blood flow to the heart muscle and it starts to become anoxic, or hypoxic anyway, low oxygen. And so you get a lot of pain in your chest. So if you relax these veins, then less blood comes to the heart. It doesn't stretch as much, and so it can get more perfusion, and it relieves the pain, decreases your ability to exercise a bit. But
So is it like with like is it a drug that's used for like normal people, or is it like people who are suffering with, like they have that problem that they
right,
cannot even go undergo like little exercise? Because like,
exactly.
okay, okay,
Yeah, that's called exertional angina. They notice that when they exert themselves, they get chest pain. And then the doctor gives them these pills that they use in an emergency, put it under their tongue, dissolves into the bloodstream really quickly, and it relieves the problem. It doesn't solve the problem, just makes the pain go away.
okay,
And then what you have to do is go in and fix the problem with surgery.
okay.
Okay, so anyway, the whole point is nitric oxide relaxes veins, and it relaxes arteries too. But in the mechanism of nitroglycerin, for some, the venous relaxation is more important. But it relaxes vessels, and that causes more blood flow. So anyway, how does this happen? Well, you've got two different cells in the blood vessel. One
Are
is the
you, sorry, are you sharing the screen or?
oh shit. I'm sorry.
Mhm.
Thanks for
That's okay.
reminding me, and I'm sorry for being crude there. It's when I'm home, I forget that I'm supposed to be professional.
That's
So I
okay.
use the S word.
I don't mind that. I actually don't mind anything. So
Okay.
yeah.
All right. So anyway, in a blood vessel, whether it's an artery or a vein, you've got an internal layer, which is the endothelial cells, and these don't contract or relax or anything. They just modulate what moves through from the blood outward. And then you've got some connective tissue, and then you've got the smooth muscle, and then more connective tissue. So we've got two different cells we're talking about here. One is the endothelial cell, the other is the smooth muscle cell. So what happens is you've got receptors on the endothelial cell. A good example is histamine receptors. You know, when you have some kind of injury or injury or infection or something, you get a release of histamine in the local area and it swells up and turns red, right? And that's because histamine relaxes the vessels, the arteries, and more blood comes in. So that's what's happening here. You've got these receptors on the endothelial cells, like, you know, histamine receptors and a lot of other receptors that'll do this as well. And these histamine receptors then will activate GQ, the G protein GQ. GQ then activates phospholipase C. Phospholipase C breaks PIP2 down into IP3. So that whole mechanism that we talked about in the class previous to this gets activated here. And it's shortcutted here in this figure because there's so much. So anyway, IP3 gets released into the cell. It binds to the IP3 receptor. You get calcium release from the ER, and that activates calmodulin. So I talked about calmodulin in the previous lecture as well. Calmodulin is a calcium binding protein that then activates a series of different enzymes. I talked about the calmodulin dependent protein kinase before. This is a different enzyme being activated by it, and it's NO synthase, nitric oxide synthase. And again, I love an enzyme that tells you what it's going to do. You know, the enzyme is named for what it does. It synthesizes nitric oxide. So NO synthase then is going to take arginine. Here is the R group of arginine. It's going to take one of these nitrogens and make nitric oxide out of it. And then citrulline is left, but we don't really care about that. That's just a byproduct. But this is nitric oxide. And you can see it's nonpolar and it's small, so it's diffusible. It'll diffuse from one cell to another right through the plasma membranes. So this nitric oxide gets produced, it diffuses into the smooth muscle cell, and then it activates the enzyme guanyl cyclase. So guanyl cyclase is similar to adenyl cyclase, except it's going to turn GTP into cyclic GMP. And cyclic GMP is going to be similar to cyclic AMP. It's going to activate a protein kinase. It's just a different protein kinase. Instead of PKA, this is PKG, protein kinase G. And protein kinase G then will phosphorylate a bunch of proteins in the smooth muscle cell and cause it to relax.
Okay.
So bottom line here being nitric oxide is another kind of second messenger, but it's almost like a first messenger too, because it comes over here. But you activate that whole IP3 calcium system, which binds calcium to calmodulin. You activate NO synthase, nitric oxide. That diffuses over here and makes cyclic GMP. And so this happens in this artery and vein system. Also happens in the brain and a lot of other places. You know, this is how cells will talk to each other and produce cyclic GMP and cause changes. I don't have any examples in the brain, but I know it's there and I know it does a lot. Okay, any questions about this?
I just have one question to back up a little. What exactly is calmodulin? Like, is it like a hormone or like what?
Let me show you. Let's back up a few slides because I talked about this. This is calmodulin. So this is actually its ribbon structure. It's got 4 binding sites for calcium. And when the calcium binds, it changes conformation, and then it binds to some other protein and activates it. And that could be kinase, or in this case, that NO synthase.
Oh, perfect. Makes sense.
Okay. Anything else here?
No, nothing else over here.
Okay. All right, so another thing that G proteins can be coupled to, like this whole section is about G protein coupled receptors and what G proteins can do. Another thing they can do is activate channels. So we don't really talk much about electrophysiology here. If you take my drugs class, and I suggest you all do, it's a really good class.
I am taking it this summer.
Oh, are you? Awesome.
Just
Awesome.
just because you've advertised it so much. So I'm like, let's see. Yeah.
Okay, so uh, in a lot in the brain, you've got, and also in the heart, and a lot of other places too, I imagine. You've got receptors that will be linked to ion channels through a G protein. So the interesting thing about potassium channels, this is a potassium channel, is that you have the same situation where the receptor gets activated. It causes the alpha subunit to release GDP and pick up GTP. The beta gamma separates from it. The difference here is it's actually the beta gamma that's going to be activating the channel, which is kind of weird. Typically, we assume that the alpha
Yeah.
subunit is the active part. Um,
Mhm.
so the other role, the other thing we talked about the beta gamma doing besides hanging out and being there is activating the GRK, G protein coupled receptor kinase, that causes downregulation of the receptor. So that's another role for the beta gamma. Mostly it's your alpha that's doing things. But here you're talking, you're talking about a hormone binding to receptor, activating a G protein, which then activates a channel.
Okay.
And another example I used in class is in the heart. The beta adrenergic receptor will activate GS, and GS will actually bind to a calcium channel and activate it directly. So that's just
Okay.
another role, but it's not that important. It's just an example to get the point across. Okay, and this is just example. Oh, growth factors. Okay, so now we're going into a totally different type of hormone receptor um, system. So all these previous ones with G protein, large heterotrimeric G proteins, were coupled to things, second messengers in the cell that kind of cause physiological changes in the cell. So smooth muscle is always my favorite example, but in smooth muscle, cyclic AMP will relax the muscle and calcium will contract the muscle. And either of those things can happen pretty quickly. Smooth muscle can contract in a minute, and in 30 seconds it will relax again. So the changes can be pretty quick, and they're always reversible. Here with these kinases, the reaction is really slow. So you hit a cell with the hormone, it can take 45 minutes to an hour and a half to see any response in the cell. And that's because these responses involve activating transcription, processing the messenger RNA, transporting it out, um, going to the ribosomes, and translation. All that stuff takes time. So these are much slower. And another difference here is these tend to change the cells permanently.
Okay.
One thing that's really common is it'll cause the cell to go into mitosis and divide. So that's why they're called mitogens. They activate mitosis. So once the cell divides, it's not going to go back to one cell again. It's a permanent change, and they also cause cells to differentiate. We talk about this a lot in developmental biology, but also later in this, like Tuesday, I was talking about early embryo development and inductions and things like that. That's this type of hormone because it's going to permanently change that cell. It's going to go from a generic cell, a generic pluripotent cell, into a less pluripotent cell, then a less and less and less, till it becomes a fully differentiated adult cell and performs a role in the organism. So that's what these things are doing. They're causing permanent changes by activating gene transcription. And so the mechanism of these is very different as well. You've got your receptors for the hormone, and the receptors, by the way, they're typically called growth factors because they're like epidermal growth factor, fibroblast growth factor, platelet-derived growth factor, nerve growth factor. They all kind of end in GF. So your growth factor receptor then has got an external hormone binding site, a single transmembrane domain, and then an enzyme here, a kinase, tyrosine kinase domain. And that's kind of interesting because most of our kinases are serine threonine kinases. These are only going to phosphorylate on tyrosines. And then in addition to having a tyrosine kinase, it's got a long tail on it that has got a bunch of tyrosines on it. So these tyrosines are going to be phosphorylated by the kinase, and that's an important part of the mechanism. So here's what it actually looks like in the cell. Once they clump together, you activate this tyrosine kinase, and then this tail comes down a bit. That's not that important. Okay, so here's the mechanism. You've got your receptor all by itself, hormone binds, and when the hormone binds, the receptors clump together. And now it brings like this kinase domain in close proximity to these tyrosines, and this kinase close to these tyrosines, and so they cross-phosphorylate each other. And now you've got all these tyrosines phosphorylated in that tail. Now these tyrosines are going to be what's called an SH2 domain.
Okay.
So SH2 comes from SRC homology domain 2. SRC is a small kinase that we talk about in development of the biology of cancer next semester if you're in that class. But basically just another kinase that's important in signal transduction. But it's got 3 domains in it, and one of the domains is very similar to this, and that's why it's called SH2 domain. These are found everywhere, and they're really important because, let me just go ahead. Here's a typical receptor, a bunch of phosphorylated tyrosines. Now, each of these phosphorylated tyrosine is in a group of amino acids called a consensus sequence. So here's tyrosine, isoleucine, tyrosine, valine. So this is going to be specific for a protein like SRC, okay? So SRC, when it sees this, will bind to that with its SH2 binding domain. So each of these has got a different enzyme that's going to be specific to it. Here's PI3 kinase, which we talked about, I forget, a different day, the PI3 kinase pathway we talk about later in the chapter. G-protein activating, GPase activating protein we talked about, so phospholipase C. So all these enzymes then get recruited to that activated tyrosine receptor, tyrosine kinase receptor. But the ones we're most interested in right now are the GRAB2 sites. Okay? Later we'll talk about JAK and STAT, but right now it's GRAB2 that we care about. So GRAB2 is kind of an adapter protein. It's got an SH2 binding domain at one side and SH3 binding domain on the other side. So that SH3 domain causes this other protein, SOS, to stick to it. So this GRAB2 is an adapter between the receptor and the SOS. SOS is a GEF, a GTP exchange factor. So what happens is once you get this, this SH2 domain forming here, that brings this whole thing up to the membrane, and now it's close to RAS. RAS is a small monomeric G protein. So different from the large trimeric in that it's monomeric. It's only one subunit. So RAS, normally when the system is off, has got GDP on it. When SOS is around, SOS causes it to release that GDP, picks up GTP, and now it's active. So this RAS is very similar to what we talked about in chapter 13 about making microfilaments with RAC-RO CDC42. This is in that family of G protein. But this one's more important in this pathway than in those other pathways. But anyway, RAS gets activated. It then recruits a protein called RAF. RAF is a kinase. RAF then phosphorylates MEK to become an active kinase. MEK then phosphorylates MAP kinase to become a kinase. So this is what we call the kinase cascade. RAS activates RAF, RAF activates MEK, MEK activates MAP kinase. So these are all serine threonine kinases. And so you can see the amplification of the signal here because each one of these phosphorylations is an amplification, So we get this big kinase cascade activating, and that activates this bad boy, MAP kinase. MAP kinase then gets translocated into the nucleus. Don't you love the way scientists can't say moves into the nucleus? We have to say translocated. Makes us sound smarter. So anyway, MAP kinase comes into the nucleus. It phosphorylates a protein called Jun. Jun is half of a transcription factor. So when Jun gets phosphorylated, it binds to the other half, which is Phos. And when Jun and Phos come together, we call them AP1.
Okay.
And then that goes on and activates cell division. And we'll talk about, like, when we get into the next chapter on cell division, we'll pick this whole mechanism up at this point and move forward and see how that actually triggers the cell to divide. But for right now, hormone binds to receptor. Receptor transphosphorylates itself after clumping together, and that produces these SH2 domains. That brings GRAB2 up. GRAB2 activates RAS, and then RAS activates RAF, and then RAF to MEK, MEK to MAP kinase. MAP kinase comes into the nucleus, phosphorylates Jun, and then you get AP1.
Okay.
So the next thing is kind of interesting in that if you were doing experiments to find out what this particular growth factor receptor does in a cell, one way to figure out what that receptor is doing in the cell is to knock it out and see what doesn't happen when it's not there, right? It's kind of like a backwards way of looking at its function. The way to do that then is to take the DNA for this and cut out a big segment of the DNA. So when it's made, you only get this extracellular domain.
Okay. And
that's what we're talking about here in this site over here. So you take that receptor, you get rid of the kinase domain. So then any FGF around is going to be bound up by those inactive receptors. So we call it a dominant negative for that reason, because you can take this, I think we talk about it Tuesday when we do development again. You can take this construct, inject it into both cells at the 2-cell stage of an embryo, and totally knock out that receptor in the embryo. And then you watch it develop and see what happens, like what's missing when you don't have FGF. And so that receptor, you're going to overexpress it. So there's more of that mutant receptor than there is the endogenous FGF receptor, the one the cell's already making. And so any FGF is either going to bind to these mutant receptors or bind between a good receptor and a mutant receptor. Either way, you totally shut down receptor activity. So in the Xenopus embryo, what that does is this. You get, this is normal tadpole, and this is the FGF negative. So you still get these anterior structures, most of them anyway. It looks like he doesn't have much of a brain, which is really important. FGF is important for brains. But you get most of the anterior structures, but you get no posterior structures. There's no tail to this thing. So from this experiment, you're like, oh, look, FGF is important for the embryo to make a tail. So those posterior structures are going to be induced by FGF. Okay, so that's the general concept of the dominant negative. And the reason why that's important is I like to ask questions like this. What would happen in the embryo if you put in a dominant negative activin receptor, or something like that? And then the way you answer that question is, okay, I know the activin receptor does this. So if this doesn't happen, this is probably what you're going to get. That's hypothesis generation. That's the way you want to be thinking about these questions of mine. Okay, so speaking of activin, we don't really talk about activin in this class, but activin is.
Yeah, I was just like, actually we did mention it, but I don't really know if we talked about a signal transduction pathway.
Okay, yeah, it's not that important. Activin itself, it's important in embryo development, but that's about it. But it's a member of a group of signaling molecules that are called TGF beta-like molecules. So TGF beta, transforming growth factor beta, is like the lead hormone of this class because it was the first one that was identified. But after it was identified, a whole bunch of other hormones are found that work in almost the same way or very similar ways. So we call this the TGF-beta-like mechanism or the receptor serine threonine kinase mechanism. So if you remember in the last one, the receptor had a tyrosine kinase in it. This is very similar in that the structure of the receptor is very similar, except the kinase domain is going to phosphorylate on serines and threonines rather than tyrosines.
Okay.
And another thing that's different here is instead of just having one type of receptor that clumps with its mate, you've got two different types of receptor. You've got the type 1 receptor, which actually binds to the hormone. And then you've got the type 2 receptor that binds to the type 1 when the hormone's around. Okay, so the hormone activates the type 1 and the type 1 binds to the type 2, and then you get a big clump of type 1 and type 2 receptors around. When that happens, when they clump together like this, you get this transphosphorylation thing again. You get a whole bunch of phosphates being added to the cytoplasmic side by these tyrosine kinases, or I'm sorry, serine kinases. And that's going to attract these guys, which are the R-SMAD, R being receptor. So I tend to call this the R-SMAD because it interacts with the receptor. And this is the C-SMAD because it hangs out in the cytoplasm and waits for the R-SMAD. So the R-SMAD then gets brought up, up to the receptor, binds to it, and then gets phosphorylated, and then it goes away. So the receptor complex activates the R-SMAD binds to the C-SMAD, and then they translocate into the nucleus, and then bind to upstream regulatory elements of the genes and turn genes on. Turn genes on or turn genes off. It depends on the cell and the mechanism and everything.
Okay, so it has to be phosphorylated R-SMAD.
Right,
okay.
right. So it's going to sit around unphosphorylated until the cell is stimulated with the hormone. Then it gets phosphorylated, binds to its co-SMAD, and goes in.
Okay.
And there are usually other proteins involved in the mechanism too. This is kind of general. We'll get more in depth in this mechanism in biology of cancer if you take that class. I'm always plugging my classes. Okay, here's another pathway. This is kind of almost like a mishmash between tyrosine kinase and the serine threonine kinase. They're pretty similar to something in the middle of the two. Very simple pathway. So here in this JAK-STAT pathway, you've got a receptor that doesn't have a kinase domain, but it's got two different kinases that are associated with it. So when the receptor gets activated, it will bring this TYK2 and JAK up to it. And then it becomes, just like another receptor tyrosine kinase, okay, because now you've got the hormone receptor complex with kinases added to it, and they transphosphorylate, and that transphosphorylation makes SH2 domains. Now, the other element here are these STATs, and the STATs have got both an SH2 domain and an SH2 binding domain, okay? So you've got your tyrosine in that, 4 amino acid sequence, and you've also got something that's going to bind to a phosphorylated tyrosine in a 4 amino acid sequence. So once the receptor phosphorylates itself, it attracts these STATs to come up and bind to it. When that happens, the kinases phosphorylate the STATs on tyrosine, they go off, and now they bind to each other. Each of them has got a tyrosine and an SH2 domain, so they bind to each other, dimerize, locate into the nucleus, and then you get transcription changes.
Oh.
Okay, I think it's a pretty simple mechanism. Like I say, the beginning part of it's just like your receptor tyrosine kinase mechanism, and then this is kind of like the TGF-beta mechanism, in that you've got two different mediator proteins that bind together and become a transcription factor.
Mm-hmm.
There are stranger transcription mechanisms, transduction mechanisms. Okay, here we are with our PI3K AKT pathway. So this is what I was talking about earlier when I said PI3 kinase is another kinase that gets activated by binding to a receptor tyrosine kinase. And what this is going to do is put a phosphate, PI3 kinase is going to put a phosphate on the 3 position of PIP2. So backing up to our this bad boy here. So backing way up, this is our PIP2. So here is fatty acids, glycerol, phosphate to inositol, and this inositol is phosphorylated on the 4 and 5 position. So this is PIP2, phosphatidylinositol 4,5 bisphosphate. So this is a substrate for for phospholipase C, which gives us IP3 and diacylglycerol. But it's also going to be a substrate for this enzyme, PI3 kinase, which is then going to put another phosphate on in the 3 position. So previously we had phosphates on the 4 and 5. Now we've got phosphates on the 3, 4, 5. So phosphatidylinositol trisphosphate. Now imagine this. Look at this. We got 2 negative charges there, 2 here, 2 here. You've got 6 negative charges all piled closely together in one little spot. That's going to stand out like a sore thumb, right? It's going to stick out on the membrane, and it's going to attract things to it. And what's going to bind to it are any proteins that have got one of these plextrin homology domains in them. So this is similar to like an SH2 domain in that you can find similar things in different proteins, and they all have the same function, which is to bring that protein up to its target. So this plextrin homology domain, you can see here, this is the yellow. The yellow is the protein, and then this is that fatty acid, and then here is the inositol with 3 phosphates on it. So in the pH domain, you've got arginine, lysine, lysine, arginine, lysine, lysine. These are all positively charged amino acids. And so they're going to bind to all these negative charges, and that's what pulls these enzymes up to the membrane and keeps them anchored there. Okay, so here's just some examples of things with pH domains. GEF, guanine nucleotide exchange factors. So you can get small monomeric G proteins activated by this mechanism. Phospholipase C, so you can make IP3 and release calcium. PDK1 and AKT, these are both serine threonine kinases. PDK1 will actually phosphorylate AKT, and we'll see that in a second. But this is going to activate these kinases as well. So that pH domain is important for activating a lot of different things. Now to reverse this mechanism, let's see what's my next slide. So to reverse this mechanism, because if you activated this and there was no way to turn it off, the cell would be cancerous. So this is reversed by an enzyme called P10. P10 then takes that the phosphate on the 3 carbon off, and then turns that PIP3 back into PIP2 or to PIP2 and turns this off. So P10 is part of this mechanism as the turn off. And again, just backing up a little bit. Oop. Where is it? Where is it? Where is it? All right, I don't see it here. A lot of these enzymes will have P10 SH2 domains for P10 as well as PI3 kinase. So it's like a feedback mechanism that gets activated at the same time as the feed forward mechanism gets activated. So P10 is an important part of this mechanism. Okay, so here is the mechanism. Growth factor receptor, as usual. We've got our SH2 domain being formed, and that brings PI3 kinase up to the membrane. And since it's close to the membrane, it starts converting PIP2 into PIP3. Now you've got 3 different enzymes that have got the pH domain on it, and they come up to the enzyme, up to the membrane. And AKT is kind of the important central, and they're all important, of course, but this is the important central one in that it's going to go off into the cell and do different things. PDK1 will phosphorylate AKT. PDK2 will phosphorylate AKT. PDK stands for phospholipid-dependent kinase. So these are all phospholipid-dependent kinases, but this one's called AKT. AKT is also called protein kinase B. So we saw PKA, PKC, PKG. This is PKB in the pantheon of PKs. Okay, so AKT now gets activated. Now, what that's going to do is kind of like a whole separate thing. So let me start over with this bad boy over here. So what's going on over here when there's no active AKT, this is active. So either this or this will be active. So in the absence of hormone, this complex is active. This is GSK3 beta, a glycogen synthase kinase beta. So it's named for an activity it has, which we'll talk about later in the insulin pathway. But more important than phosphorylating glycogen synthase, it phosphorylates beta-catenin. Beta-catenin is an important transcription factor that will activate cell division. Normally, when you've got no hormone there, so the cell's not stimulated to divide, you don't want beta-catenin around because it's not being stimulated. So normally GSK3 beta, along with these other proteins, are going to phosphorylate beta-catenin, and phosphorylated beta-catenin then gets ubiquitinated and destroyed by the proteasome complex. So on a regular basis, we're breaking down beta-catenin. So its level is really low in the cell normally. And these proteins, APC, AXIN, and WTX, are holding GSK3 beta and keeping it in the right conformation that it can phosphorylate beta-catenin. And that's important because we'll see that come into a different mechanism later, the Wnt mechanism. Okay, so this we're calling the destruction complex because it destroys beta-catenin. So when the hormone's around, you get the production of PI3 kinase, activation of AKT. AKT then phosphorylates GSK3 beta and inhibits it. Once GSK3 beta is inhibited, beta-catenin no longer gets phosphorylated and it builds up in the cell really quickly. So it then stimulates cell division through mechanisms that we'll talk about in the next chapter. Okay, so that's the basic pathway. Receptor, PI3 kinase, AKT, GSK3 beta, beta-catenin. All right, what's next? So here is the insulin receptor mechanism, which is going to be very similar to the AKT mechanism. So the insulin receptor, so what, the way you want to think, you know what insulin does, right? It decreases your blood glucose, right? This is how it does that. So this mechanism is really active in muscle cells and in the liver, where you're going to accumulate glucose when it's high. So glucose gets high, insulin gets released, you activate this mechanism, and this mechanism puts puts glucose transporters into the cell membrane, okay? And now you're transporting glucose into the cell, and it also activates glycogen synthase. So you're making glycogen as that glucose comes in. You don't see it in this figure from your book. Let me try something. We are Santa cells, filling Santa shelves. We are Santa cells. No, not this. Where is it? Sorry, I'm trying to find an animation that I made about this. Oh, here it is. Okay. I showed this in class, and it's, I think, a lot better than that static figure from your book because it shows everything. So just take a second to come up. Come on, baby. Here it comes. So this is the program I use to make these animations. And this one I made years ago, and it's not supported anymore. So I can't put it on the web. It's made with Flash, and the Flash add-on is no longer used by Chrome or any other thing, so it doesn't really work. But I can still play it through the Adobe Animate program, so that's what I'm going to do. I got a very needy dog here next to me. I have to keep petting him. Don't I, buddy? He's needy, but he deserves. It's taking for god awful ever. All right, let me go over the mechanism as it is, and we'll see if that thing will start for us or not. Where was it?
I'm sorry, I was mute all this time. What's his name? What's your dog's name?
This is Gus.
Gus.
Gus
Okay.
the Wonder Moose. He is a golden retriever, and he's 120 pounds.
Oh, I love golden retrievers. My best friend has one, and his name is Leo.
Leo, that's a great name too. Yeah. Goldens are just the sweetest dogs. I can't imagine him ever biting anybody. He loves people.
Okay. They're just so innocent.
Yes. Okay, let me go through this, and then hopefully that animation will come up. So when insulin is around, the receptor clumps together, just like any other RTK. The only difference is the insulin receptor has got two different subunits instead of having it all in one subunit. Okay. Once that activates, it produces these SH2 domains, and that brings up insulin receptor substrate 1. So that comes up and gets phosphorylated as well. And then PI3 kinase binds to that. And the PI3 kinase then makes PIP3. That activates PDK1, PDK2, which then phosphorylate AKT, which becomes active. And AKT then phosphorylates a protein on these vesicles that are just subcellular, and that causes them to fuse into the membrane and puts those glucose receptors into the membrane. And then here...
I'm sorry, are those glucose receptors or transporters?
Transporters. It's called GLUT4. So it's one of the glucose transporters. And then AKT will also phosphorylate GSK3 beta. And here in these cells, GSK3 beta is phosphorylating glycogen synthase and inhibiting it. So once AKT inhibits that, then glycogen synthase becomes active. And you're transporting glucose in and turning it into glycogen. Oh. Let me see if that thing started yet. Come on, you. It's taking a stab time. Aha. Okay. All right, let me share this. All right, because the coding in it doesn't work anymore. When I start this, it's just going to play. I can't control it, but we'll watch it happen anyway. Okay. Okay, so down here you can see glycogen synthase kinase phosphorylating glycogen synthase. Up here, hormone hit receptor, IRS is phosphorylated. We start making PIP3. That brings our enzymes up because they have pH domains. PDK2 and PDK1 phosphorylate AKT. AKT then phosphorylates that protein there, and that vesicle is going to fuse. And now we're transporting glucose in. Glycogen synthase is going to make glycogen and store that glucose.
That's a really cool animation, honestly.
Oh, thanks. You know, I was just thinking, I think maybe I could just replace the coding and it would work again. And I wouldn't have to do all the animating again. Let me just play it again and let you watch it without me talking. Okay. So I'll see if I can get that uploaded this week. And we'll get back to this. Okay, so that's the insulin receptor mechanism. It's kind of taken you a step further than the previous mechanism, but it's very similar. And then one more pathway, because I know I could see it. I was reading your face, and you're like, another pathway, Dr. Slish. Give me another one. I need another one. So here's the Wnt pathway. Now, this is kind of related in that we start in the center about the same as we did in the PI3 kinase pathway. This is your destruction complex, and it's phosphorylating beta catenin, and it's getting broken down. So and we've got the same proteins circling it here. So that's kind of the center of this pathway as well. The only difference is that we're going to inhibit this destruction complex in a different way. So Wnt is a hormone that's really important in early embryo development. I mean, you can't get an embryo without Wnt. It's so important. So Wnt, then when it's around, will bind to its receptor, which is frizzled. And when Wnt binds to frizzled, this other kind of co-receptor comes in, this LRP, and they make a complex that then attracts disheveled. And disheveled comes up, and it pulls axin off the destruction complex. And without axin, GSK3 beta becomes inactive. And just like we saw before, when that shuts off, beta catenin builds up, and then it translocates to the nucleus and binds to TCF-LF, which then becomes a transcription factor. So this whole part of the process is very similar to the AKT mechanism. The only difference is you've got disheveled, frizzled Wnt and LRP at this side that's controlling it. And Wnt makes this complex form, which then pulls axin off, and that allows beta catenin to build up. Okay. I believe that's it. Yep, that's it. So do you have any questions? Oh, you're muted again.
Oh, yeah. Nothing. I was saying, yeah, we actually covered 2 lectures.
Oh, did we?
Yeah.
Rock and roll. 15 minutes, I went through it all.
Yeah, but that was great. I understood it. I just have one question. So, when are you planning to take like the exam, the next exam? Is it like, I think it's on like April 13-ish week, but I don't know if you're planning to like move it ahead or.
Well, next week on Tuesday, we're going to finish development. And then on Thursday, we're going to have a review. And then the following Tuesday, we'll have, so whatever dates that is, it's like a week from this coming Tuesday.
Okay, perfect. And also another thing that I wanted to ask you, like for the final exam, okay, one before that. So the lab, is it 40% of the grade?
It's 25%.
Okay, it's 25%. Okay. And then like for the final exam, which is going to be of 200 marks, like is the pattern going to be similar? Like how is it going to be like? Is it going to be like actually like double what we are doing in like our normal exams?
I think there's an example up there, but typically
Okay.
what I do is I'll give you, let's see, how many chapters are there? 2, 4, 6, 8. 8 chapters. So I'll give you 16 questions and tell you to answer 8 of them. And they'll all be the
Okay.
kind of straight out of the notes questions, not the more complicated ones, but the easier ones.
All right. That's great.
Okay.
Thank you so much for making our life easier.
Oh, sure. No problem. Let me tell you one more thing before you go, along with everyone else, anyone else who decides to look at this. I haven't put it up yet, but in Moodle, there's a game that I'm going to put up like now, as soon as I hang up with you. And it's only on signal transduction, not the development part. So you can play that anytime you want. Okay.
Okay. Is it for bonus points?
Yes. 10 bonus
Okay.
points. Yeah.
Okay. But is that excluded from the exam 3 bonus game, or is it the same thing?
It's the same thing.
Okay.
Yeah. It's exam 3 bonus. Okay.
All right.
Okay.
Thank you so much. Have a great day.
You too.
Bye-bye.
Bye bye.